this section, we'll be looking at stage 1 of the third line of defense, acquired specific immunity. Before we can look specifically at lymphocyte development, we need to review some of the unique structures of molecules and characteristics of cell surfaces, like the membranes and the envelopes, and gene expression. The cell surface characteristics are responsible for specific immune recognition and immune reactions, so it's really important for us to get a good grounding in this. And I must admit, it's some pretty complex stuff, but hang in there, spend some time with it, I promise you'll get it. So, a given cell can express several different types of cell markers. Each has a distinct role in detection, recognition, and cell communication. The major functions of the immune system markers are attachment to non-self or foreign antigens. They could be for binding to cell surface receptors that indicate self, such as MHC molecules. We'll spend some time covering those. Receiving and transmitting chemical messages to coordinate the immune response, and aiding in cellular development. Now, MHC proteins, these are major histocompatibility complexes. It's a set of genes that codes for human cell receptors. They give rise to a series of different glycoproteins called MHC molecules that are found on all cells except for red blood cells. They are also known as human leukocyte antigens, or the HLA system. Anyway, there are 3 classes of MHC genes that have been identified. The class 1 genes code for markers that display unique characteristics of self. This is a complicated system, but the general idea is that each human being inherits a particular combination of class 1 MHC genes. In general, the closer the blood relationship, the greater the probability that you have a similar MHC profile. However, it's unlikely that even related people will have the exact same profile, just because of differences that develop between individuals. Class 2 genes code for immune regulatory receptors that are found on macrophages, dendritic cells, and B cells. They are involved in presenting antigens to T cells during cooperative immune reactions. And then the class 3 MHC genes encode for proteins that are involved with the complement system. We've already covered the complement system, and we won't be looking at the class 3 genes. The focus in this section will be on class 1 and class 2 MHC genes. Let's take a closer look. MHC proteins, again, are found on host cells. If they're absent, the cells will be destroyed by the immune system. Generally, these MHC complexes are used to present different antigens. MHC1 molecules are found on all nucleated human cells. And then MHC2 complexes are found on only some types of white blood cells. As we go through the rest of this chapter, you'll see how these come into play. So both B cells and T cells have receptors on them that bind to antigens. The B cells have receptors that bind antigens directly, whereas the T cells have receptors that bind processed antigens that have been complexed with MHC molecules. So check this out. Remember that the antigen was reaching B cells just by floating through the lymph node, whereas it was reaching T cells because a dendritic cell picked it up and carried it and presented it to a T cell. So here we've got the dendritic cell now carrying antigen, and it's displaying it to the T helper cell. So that's all we're saying here, is that the T cells are presented antigens, whereas the B cells will bind directly to the antigens. So it takes an antigen presenting cell, like the dendritic cell, to present the T cells with antigens that have been complexed with an MHC marker. Simply, the red and the green here is that complex, whereas here the B cell is receiving just the antigen, not bound to any kind of MHC protein. So now let's look at some specific events in B cell maturation. We know that B cells are maturing in the bone marrow. And the bone marrow harbors stromal cells. Stromal cells are huge cells in the bone marrow that nurture the lymphocyte stem cells and provide chemical signals that initiate B cell development. Once the B cells have matured, they'll circulate through the blood, homing in on specific sites in, say, lymph nodes or spleens or GALT tissues. Once they arrive there, they adhere to specific binding molecules, where they'll eventually come into contact with antigens throughout their lifespan. Now, T cells have a different situation. We know that T cells are matured in the thymus gland, and their development is directed by the hormones of that gland. All mature T lymphocytes express extra surface proteins. They have a CD3 co-receptor, as well as either a CD4 or CD8 co-receptor. Don't worry about what the names mean. The CD4 co-receptors bind to MHC class II markers and are expressed on T helper cells. CD8 binds to MHC class I molecules and are found on cytotoxic T cells. Don't worry about the details of this yet. It'll all make sense as we move through the chapter. Either way, just like the B cells, the T cells constantly circulate between lymphatic and general circulatory system, and they migrate to specific T cell areas in the lymph nodes and spleen. So the lymph nodes and spleen have T cell areas and B cell areas that are separate. This table will be a great tool to come back to. It summarized some of the different properties of B and T cells. For example, as I was just saying, where are they in the different lymphatic organs? The B cells are found mostly in the cortex areas, whereas T cells are found mostly in paracortical sites. The B cells have immunoglobulin cell markers, whereas T cells have the T cell receptor and then those extra CD molecules. So onto the idea of the immunoglobulins. How do we get so much diversity in immunological structures? Because each B and T cell is specific to only one antigen. So we need to have millions of different B and T cells, maybe even billions of different B and T cells, leading to that sort of specificity. And each one of them is produced without even seeing any exposure to an antigen. So they have to come up with literally billions of different receptors just in case a molecule comes along that fits their receptor. So by the time B and T cells reach their lymphoid tissue, they're equipped to respond to just a single and unique antigen. Now there's some mystery as to how this happens, but we have some insight. There are 500 different gene segments that can be rearranged to produce these diverse receptor types. Again, each receptor has to be specific to any possible antigen that might invade the body. So how does this possibly happen? Well, early undifferentiated lymphocytes undergo a continuous series of divisions. During these series of divisions, there are many genetic changes that occur. And how do these genetic changes occur? Well, let's look at a generalized receptor. The generalized receptor looks like this. It has two heavy chains and it has two light chains. So the heavy chains are the long chains on the inside and the light chains are the shorter chains on the outside. And again, to review protein production, we have DNA that's then transcribed into messenger RNA and then translated into the end product, these proteins. Here are the gene segments. And there's a number of different choices of genes that could be taken from the long chain segment and the short chain segment. So in this case, we've got one from over here, one from over here, and one from over here coming in to produce this short chain. Because of the shuffling of these different pieces, we can have hundreds of million different types of B and T cell receptors, so that each cell can have a unique receptor region. The actual receptor region is right here in between the two short chains. The very tips of both the heavy and light chains are variable segments. That's where they have great genetic differences, so that each site is distinct to a particular antigen molecule. Let's watch a quick animation so that you can understand this a little bit more clearly.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chains of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters, designating IgG, gamma, IgM, mu, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced.
So you don't need to know all the details of that, but hopefully that gives you an idea how we acquire so much diversity in these antigen receptors or antibodies. So let's review quickly this lymphocyte development, because I know that's a lot of material. The lymphocyte starts out in bone marrow and becomes either a T or a B cell. The B cells stay and develop in the bone marrow, and the T cells migrate to the thymus to continue their development. They build their unique antigen receptors while they're in their respective maturation zone. Then they migrate to secondary lymphoid tissue. So all of that diversity of receptor production or antibody production occurs before they migrate to their secondary lymphoid tissues. So throughout that process of early development, we have a whole repertoire of clones, each with a unique receptor display. The second stage of development requires stimulation by an antigen, let's say a microbe. When the microbe enters the immune surveillance system, it encounters all of these different lymphocytes. One of them is specific to that antigen and is ready to recognize the antigen. The antigen contact with the lymphocyte stimulates that particular clone to undergo mitotic divisions. This expands the population, creating a large population of lymphocytes that all bear the same specificity. Let's watch a quick animation of this process.
The immune system can produce antibodies against millions of different antigens. B cells, which are constantly produced from lymphoid stem cells, are found in the blood and lymphoid tissues. Each B cell can synthesize only one of the millions of possible antibodies and displays this antibody on its surface. When an antigen meets a B cell having a surface antibody of the proper specificity, it complexes with the antibody. B cells that are not activated do not develop further. Stimulation of only the B cells that carry antibodies that react with antigen is referred to as clonal selection. Following antigen-antibody interaction, antigen-antibody complexes aggregate on the surface of the cell. This is called capping. The antigen-antibody complexes are then internalized. The activated B cell swells and begins to divide rapidly, producing a B cell clone. The B cells then differentiate into plasma cells and memory cells. Plasma cells produce the specific antibody that provoked its formation, whereas memory cells remain in circulation but do not produce antibodies. Memory cells can become activated by a later challenge by the same antigen. Many antibody responses require signals from T helper cells. The T helper cell is stimulated by an interaction with an antigen-presenting cell such as a macrophage. The macrophage ingests the antigen, digests it, and presents it on the cell surface via a class II MHC. The macrophage then activates a T helper cell that carries a T cell receptor capable of recognizing the antigen on the class II MHC of the macrophage. The B cell presents the antigen on its surface using class II MHC. The activated T helper cell interacts with the B cell and secretes chemical signals called interleukins to stimulate the B cell to differentiate into a plasma cell and produce antibodies.
So hopefully this animation gives you a better picture of what's going on during lymphocyte development. So in the production of all of these different receptor molecules or antibody molecules, why is it that some are not created that react to self? And it turns out that there's a process of regulation that occurs during development. So any clones that react to self markers are destroyed through clonal deletion. So these cells whose receptors react to self are deleted, and so they're no longer in this group of lymphocyte clones. It's thought that autoimmune diseases are caused by loss of this immune tolerance to self, so lack of the control and ability to eliminate self-attacking clones. So now we have an understanding of lymphocyte development. Let's take a closer look at the differences in receptors before we move on. So B cell receptors are immunoglobulin molecules, or IGs. The receptor genes that undergo recombination are those that are governing immunoglobulin synthesis. IGs are large glycoprotein molecules that serve as antigen receptors for beta cells and as antibodies when they're secreted. They have the Y-shaped arrangement with the heavy chains and light chains. The ends of the forks contain pockets, which are the antigen binding sites. Each individual immunoglobulin molecule has a different binding site. Again, these immunoglobulin molecules are receptors on the B cells to antigens, and they're antibodies when they're released from the B cells. They can be highly variable in shape to fit a wide range of antigens, but the classic one has this Y shape. Again, there are variable regions and constant regions. Whereas the T cell receptor has a little bit of a different shape. It's never actually secreted, so they're only on the surface of T cells. They too have a variable region and a constant region, as well as their antigen binding site. Again, on T cells, we have both CD3 receptors as well as the antigen binding receptor, and either a CD4 or a CD8 receptor. Before we close up this section, let's have a quick concept check. Where do T lymphocytes complete their development? Oh, this one's an easy one. There's 3 T words we don't want to get mixed up: thyroid, thymus, and thalamus. It's definitely not the hypothalamus, or the thalamus, or the thyroid. Definitely the thymus, located right between the lungs and above the heart.